In this chapter, we will explore the technical nature of classifying dinosaurs. To understand the features used in the classification process, we'll have to go over some important anatomical terms. If it wasn't obvious, the term anatomical just means relevant to anatomy. We, as well as dinosaurs, are vertebrates, animals with spines made of vertebrae. All vertebrates inherited this trait from our common original vertebrate ancestor. Primitively, our ancestral vertebrae were not fused together to form a single solid structure surrounding our spinal cords. Instead, they began as unfused units at each segment. The thimble-shaped lower portion of our vertebrae that stack and support the compressive forces of our weight are the centra, or singularly, each is a centrum. The top of our vertebrae that bear the long spines and processes for attaching muscles and ligaments are neural arches, and the spine at the very top of each vertebra is a neural spine. The hole formed where each centrum and neural arch meet is a neural canal, and this is where the spinal cord of the central nervous system passes through from our brain down the vertebrae to the vertebrae between our hips. Just as the individual parts of each vertebra has names, different vertebrae within the vertebrate skeleton have names based on where they occur. The terms denominating each variety of vertebrae are interchangeable with the region of the body, so tail vertebrae are called caudal vertebrae. Caudal means anything relevant to the tail, or even the direction of the tail. So if you were describing the path of the spinal cord, for example, you would say that, beginning from the brain, the spinal cord passes caudally through each vertebra, to use the term in its orientational or directional sense. The opposite of caudal as a direction happens to be rostral, meaning toward the rostrum, and rostrum is another word for the nose and everything close to it. So rostral to the caudal vertebrae are the vertebrae between the hips. In most advanced vertebrates, at least some of these vertebrae are fused together, and these fused vertebrae between the hips form a sacrum. The vertebrae forming that sacrum are appropriately called sacral vertebrae. The vertebrae of the back are called dorsal vertebrae. You may have heard of dorsal fins, maybe while enjoying some Shark Week programming. Dorsal refers to everything having to do with the region of the back, as well as the direction of the back relative to the belly, or the ventral side of vertebrates. You might not have heard your own back vertebrae called dorsal vertebrae before, but instead may have heard the terms thoracic and lumbar vertebrae. Mammals are weird, and this is probably not the first time you'll hear me say this. Unlike reptiles and birds, and just about every other group of vertebrates, we only have ribs attached to some of our back vertebrae, and almost never have any on our neck vertebrae. So we designate which of our dorsal vertebrae do and do not have ribs. Our lower back, which is rib-free, is composed of lumbar vertebrae, while our upper back, where our ribs are, is composed of thoracic vertebrae. The neck is the cervical region of the vertebrate body, so its vertebrae are cervical vertebrae. We also have a couple of specialized neck vertebrae, the two closest to our skulls. The atlas vertebra is the first cervical vertebra, the one that attaches to the occipital condyle of the skull. The second cervical vertebra is the axis vertebra. In addition to these general regions of the body and the vertebrae that occur there, there are some other key orientational terms that you should become familiar with. The axial skeleton is the part that forms the trunk of the body, the skull, and the tail. The appendicular skeleton refers to the appendages, arms and legs. The bones of the appendicular skeleton all have one end that is closer to the body's center and another that is relatively farther away. The near end of bones is the proximal end, which you can think of as being in closer proximity to the center of the body. The far end is the distal end, which is more distant from the center. The appendicular and axial skeletons are joined at the body's girdles, the hips, or pelvic girdle, and the shoulders, or pectoral girdle. Vertebrate hips are comprised of 3 pairs of bones, a right and left pubis in the pubic region, a right and left ischium, which are what we sit on, and a right and left ilium, which are the bony knobs that our belts rest on when they are holding our pants up. The socket formed at the juncture between these 3 bones is where the head of each femur articulates, and the socket is called an acetabulum. The pectoral girdles are formed of scapulae and coracoids in dinosaurs. This is a little different from our own arrangement of scapulae and clavicles, a result of our 2 lineages' different evolutionary histories. We do call our shoulder sockets the same thing, though, that being glenoid fossae, or singular glenoid fossa. If you have not had this epiphany before, you're probably noticing the bones of dinosaur skeletons are the same bones that make up our own skeletons. They are just shaped and sized differently, and in some cases we may have different numbers of things like fingers, toes, or body segments in the neck, back, or tail. As I said when I was explaining how our vertebrae are named and arranged, this is due to our common evolutionary history and the inheritance of traits from generation to generation. Approximating this evolutionary history has become the goal of the technical classification process. The way we group organisms and the models we build of these groupings is all made to represent the relationships and evolutionary history of each species and of all species. The formal practice of unraveling these relationships is currently done using the complex mathematical process of cladistics. To perform cladistic analyses, the physical and behavioral traits of organisms are coded into a matrix. This is often a sort of binary quantification of qualitative data, like the presence of a particular trait is denoted as a 1 and its absence as a 0. It does not have to be binary, though. For example, the ratio of 2 bone lengths compared to one another might be coded 0, 1, or 2 if the ratio is below, within, or above a certain value. Once coded, the matrices for all of the species being compared are analyzed for the most likely evolutionary history, the history that would result in the combined array of traits observed across the species being compared. Mainly what the algorithms are trying to mathematically tease out of the data for 4 basic kinds of ways to categorize each trait of each species. Each trait should either be 1, a plesiomorphy, which is a primitive trait, 2, a synapomorphy, which is a trait that evolved at some point within the evolutionary history being analyzed and is shared by 1 or more species, so it is a shared derived trait, 3, novelties, which are traits that only occur in one of the clades being analyzed, or 4, an example of convergent evolution, which would be a trait that is shared but was independently derived, rather than evolutionarily inherited. These traits are passed down from one generation to the next through an organism's genes. Genes are contained in the chains of amino acids that make up our chromosomes. The location of a gene on its chromosome is its locus. In ourselves, and we presume dinosaurs, chromosomes and the genes they bear are paired with each gene having a copy at each locus. Each variation of a gene is an allele. When there are different alleles at the same locus on the chromosome pair, the dominant gene determines which is expressed as the organism develops, and its phenotype becomes apparent. You've probably heard about the gene pool and jokes about who came from the shallow end. Gene pools can be thought of as the collective genetic material of any given population. The appearance, fitness, fecundity, etc. of a population is determined by the alleles available to be passed down to the next generation. This is the genetic material with which natural selection has to work in driving the evolutionary process of a population. Natural selection, along with mutation on a microevolutionary scale, shift the relative frequencies of these alleles within a population. This shift is microevolution. When enough changes accumulate to constitute the erection of a new species, that is full-on evolution, with no prefix necessary. When more extreme shifts occur, above the species level, these are referred to as macroevolutionary events. Macroevolution tends to happen in bursts, often resulting from a major change in climate or an extinction event wiping out a significant chunk of biodiversity and leaving a bunch of vacant niches to be filled. This bursting pattern, observable in the fossil record, is referred to as punctuated equilibrium. Stephen Jay Gould and Niles Eldredge brought this evolutionary concept to light. It countered the widely held perception that evolution happened like plate tectonics occurs at a slow and steady gradualist pace. What we see instead, for lengthy periods, millions of years at a time, with little change, whole, stable ecosystems persist with few, if any, species evolving. Then, in the blink of a geologic eye, we see massive turnover in the species that fill the major niches and that even carve out new niches. Niches are not to be confused with things we call niche or niche, which is used to denote things catering to a narrower, more specialized band of customers. Niches in the biological sense are often specific, but an organism can fill a niche that is being a generalist. Cockroaches are an example of an organism that can adapt to a variety of conditions and nutrition sources, so their niche as an insect genre could be considered a broad niche, or perhaps they're specialized at being generalists. An organism's niche is not just defined by how it gets its food and water. Its every interaction with its ecosystem is part of its niche. Where a species sleeps, which other species it competes with for resources or territory, even the earth it may dig up and the fecal matter it may leave in that hole are all facets of the niche an organism fills. Thinking about the interconnectivity of ecosystems makes this seem more tangible. Somebody has to supply the invertebrates that rely on feces for nutrition, their resources. And as unexciting as it may be, this, along with everything else, is part of an organism's niche. A pair of species can even drive each other to rapidly evolve some new extreme features, like predator and prey driving each other to continually increase their potential for running at high velocity, or their massive size. Such a relationship is sometimes called a Red Queen scenario, likening this evolutionary arms race between species to running on an accelerating treadmill, so that you have to keep running faster to stay in the same place. This interconnectivity is also what causes the ripple effect an extinction can have. If one species gets wiped out suddenly due to disease, drought, famine, or whatever, the other species that relied on it for prey, or to prey on its predators to provide it with shelter or provide it with feces to eat, such a vacancy can quickly cause a chain reaction and the collapse of an ecosystem. Organisms are honed by natural selection to be fit to their complete ecosystem. Natural selection does not have a mechanism to prepare species for changes it may face in the future. Having enough genetic diversity present within a population so that enough individuals survive and can adapt to changes is the biggest trick to being a species with longevity. Tracing the macroevolutionary changes of vertebrates through time is how we are able to determine where dinosaurs fit on the proverbial tree of life. As you may have imagined, if we look back far enough, we arrive at the common ancestor between our own mammalian ancestors and dinosaurs. People and dinosaurs are both vertebrates, and more specifically, we are tetrapods, so that places our common ancestor above or more recent than the split between ray-finned fishes and lobe-finned fishes. We know also that dinosaurs laid eggs like amniotes, and more specifically, they were oviparous, laying eggs that hatched outside the mother's body. As mammals, we too are amniotes, but humans are viviparous, retaining fertilized eggs and developing embryos internally, embedded in a womb. Knowing for certain how a fossil organism handled its embryo incubation can be tricky to downright impossible to find out. Thankfully, the evolutionary relationships of tetrapods seems to have been recorded in the evolutionary development of their skulls. Modern reptile skulls come in two basic varieties: anapsid skulls, which turtles have, and diapsid skulls, which everything but turtles have. Our own mammalian lineage springs from the other branch of amniotes, which primitively have synapsid skulls. These three skull types are defined by the number of fenestrae, or windows, that occur posterior to the eye sockets. Our synapsid ancestors, like the famous sail-backed Permian synapsid, Dimetrodon, have one hole behind the eye, so Dimetrodon is not a member of the Eureptilia. The true reptiles, which is what the Eureptilia are, in the late Triassic spawned dinosaurs and pterosaurs from a group of diapsid reptiles known as archosaurs. Archosaurs are diapsids, with a fenestra anterior to the eye, in addition to the two that are posterior to the eye. Knowing how these major groups of reptiles are related allows us to project behavior and physiology of modern animals onto these extinct life forms. This is sort of an indirect method of observing how these animals that lived millions of years ago functioned.